all, to thank the organizing committee for giving me this opportunity and to present before my old and current tutor. Let's start. What's going on? Okay. So let's start with the more general concept. When we before talking about Cocaine syndrome, I would like to give you an overview about rare genetic disorder in Tunisia. We all know that a rare genetic disorder, it touches a very minor frequency of patients, and that is less diagnosed, and there is very few interest about this kind of subject. But in Tunisia, given the high rate of consanguinity, we observe more and more rare genetic disorders that are emerging. And so we emit this kind of hypothesis, given the fact that some rare genetic disorders share some specific characteristics that are also observed in common disorder, and given the fact that it is caused only by genetic disease, why not investigate what's common between the frequent disorder and a rare genetic disorder in order to discover new potential biomarkers? So in this work, I would like to give an overview about the genetic disorder touching DNA repair. And in particular, we were interested in nucleotide excision repair disorder. And in this disorder, there is some specific genetic mutation touching two proteins called CSA or CSB. This disorder gives emergence of a very rare and rare disorder that give a disease called the Cocaine syndrome. And this disorder is touch approximately 2.7 per million births in West Europe. But in Tunisia, we are observing more and more patients suffering from this disease. And this is characterized by growth delay. The patient suffers from many manifestations that make them more similar to old persons, and with some sensitivity and neurodegeneration. And what makes this disorder more complex is the fact that the same genetic mutation could cause this disorder or a simple other disorder that called UV sensitive syndrome. And even with the fact that both these disorders are caused by mutation in CSA or CSB, the latter patients are only suffering from minor UV sensitivity. And when we explore these two cases, we observe that for the first one, we don't have a cure, and we have a progressive neurodegeneration that leads to death at almost 8 years, while the other can live normally with UV protection. And the difference between the two is regarding the oxidative stress process. And when we explored the literature, we found that in the Cocaine syndrome patients, there is alteration in the oxidative stress process that leads to this enhanced observation in manifestation in many organs. So in our lab, as you can see, this is the photo from our lab. We out to develop specific cellular primary culture model from muscle biopsies and skin biopsies in order to research from biomarker related to this accelerated agent, and in order to, in the future, why not develop a new therapeutic perspective. And from this skin biopsies, we do the culture of fibroblasts. And first of all, we can pass out from the NGS analysis and do some specific functional analysis by stimulating these fibroblasts with UV light and measure the reparation of their DNA to confirm if they are having this syndrome or other genetic manifestation. And for example, in this specific case, we observed that even when the patient doesn't express reduced light sensitivity, photosensitivity, they have the same defect in the DNA reparation process after UV exposure. And also from this skin biopsies, we try to understand how this alteration in oxidative stress acts by exploring the gene expression of a large set of genes. And we observe that there is alteration in the oxidative stress pathway in normal condition without exposure to H2O2 or other pharmacological compounds. And what was interesting to find that there is a specific form that normally clinically we could not distinguish from the other form. And via this gene expression analysis, we found that this form have a particular way to that the genes are altered. So we further analyzed the alteration of gene expression analysis via different bioinformatical and transcriptional analysis. And we found two major altered pathways, one that touched the arachidonic acid metabolism, and the other touched the nuclear factor, erythrocyte factor NRF2. And in these patients, we have that the expression of this gene are completely repressed in a way that kind of similar to aged population. And when we try to find a specific pharmacological compound for this gene, we found that what was interesting that the nicotinamide, which is just a water-soluble active form of vitamin D3, can be specific to these genes. And recent research suggests that the supplementation with this particular vitamin could enhance and regulate some kind of oxidative stress in mouse model and also in other accelerated age genetic format. And so we try to add this region to our fibroblast culture. And what we found that the fact that we add this nicotinamide kind of correct this altered gene expression. Besides that, we try to find other genetic related compounds, and we observe that the supplementation with nicotinamide correct also some altered inflammatory genes and even some mitochondrial functioning that was not previously investigated. And to sum up this part of our work, we proved that via nicotinamide, we could some kind of decrease the level of oxidative stress that Chikhaoui syndrome are suffering from by targeting inflammatory pathway and also other oxidative stress related pathway to mitochondria and mitophagy. And in our second section, we try to think about other cellular models. So when we think about the aging in general population, we have the sarcopenia, which is the progressive decline of muscle mass and strength. And in patients with Chikhaoui syndrome, we have a muscle atrophy that we don't know how it is caused by a simple DNA repair defect. And so we also try to explore a large set of genes and to validate it. We observe that there is an overexpression of many inflammatory and fibrosis key genes that indicate a low grade of inflammation associated with atrophy. And so with our second model, we tried from muscle biopsies to isolate satellite stem cells and to induce their proliferation in vitro, and also to investigate the effect of our molecule. And via this way of thinking, we just started with an aged cellular model, and we found that via nicotinamide suppression, supplementation, we observe that the fusion index, which reflects the regeneration of muscle, is enhanced by this supplementation with the overexpression of other key genes related to muscle regeneration. And so to conclude, our study represents the first multi-parametric approach to study rare genetic disorder to study a rare genetic disorder model to study the regular aging and the altered oxidative stress genes may differentiate between two different forms that were not clinically distinguishable. And we discover a good number of biomarkers that need further investigation, and which I would like to highlight more, that several questions remain open concerning the physiopathology of many genetic disorders that are common in Tunisia due to the high rate of consanguinity. And finally, I would like to thank all those who supported our work, especially the X4 in our lab. And thank you for your attention.
Thank you very much, Asma, for this wonderful presentation and wonderful work. I really loved your work because of two things. Because for my PhD studies, I worked a lot on consanguineous, on rare disorders, so it's very close to my heart topic. The second thing is that I don't know if you watched actually the movie, The Curious Case of Benjamin.
Yes, one of the reasons that I was interested.
Yeah, so it immediately, when I saw the presentation, it immediately reminded me of that movie. So maybe, I mean, the movie, for the ones who doesn't know it, so Brad Pitt was born old and he was getting younger. Yes, it's a Brad Pitt movie, of course, so everyone will get it. So he will get younger as when he was supposed to be getting older. So yeah, hopefully maybe one day you using gene editing or I don't know, maybe we can find a solution for that. But thank you very much for the presentation. I think Asma is ready to take any questions. Yes. Please.
Thank you so much for this amazing presentation. So I want to just clarify something in my head. So the determination of the syndrome, like to know if someone is suffering from it, it stands on the inability of gene repair and regulation of possible alteration and mutations.
So the general diagnosis, usually it was using genetic analysis to find if there is a specific mutation, but there is also another variable that uses the capacity of repairing DNA after UV stimulation. So it's like that.
So it's also a suggestion, maybe. Given the fact that you see this effect when you add the nicotinamide, basically restoring the NADH depletion and everything, have you checked if in your model there is a correlation with the hyperactivation of a protein, which is PARC1, which is involved in DNA repair system? Have you ever checked if your model got this hyperactivation? Because there are lots of papers that the nicotinamide is actually very effective in.
There is some work that was done in mice model, but we didn't done it on our model, so it's a terrific suggestion. We will try it soon.
Try.
Yeah, thank you.
Thank you.
Thank you for your lovely presentation. I want to know, can stress affect osteosection?
I didn't understand. Quite affect
what? Osteosection.
C-section. Yes, C-section, césarienne.
I don't have the answer. Because usually these patients have not life expectancy to give birth, so we don't have.
You can attend from
Stress.
It's a monogenic.
Stress.
I didn't work on this.
Okay.
Wait. Another question? So we have time?
Yes, we still have time. Have a minute.
What is the comparison between the elderly and the mechanism from between elderly and the
Cocaine syndrome.
this syndrome? Because the oxidative stress is let's say response by many other pathways.
And
So you focus in comparison between elderly and patient. That's all.
in fact, we know that in elderly, the oxidative stress is caused by epigenetic other environmental problem. But in cocaine syndrome, we have only this mutation that cause this alteration in oxidative stress. So compared to the regular aging problem, there is only one factor that causes this oxidative stress alteration. So maybe when we limit that, we could discover other pathways that were not, we could not discern due to these different factors that causes the oxidative stress alteration in aged population.
So just the genes involved in
that are common with the regular aging.
Thank you, Asma. Merci. So, okay. Another talk about venom from Pasteur Institute. Actually, we have a very, for the ones who doesn't know it, so we have a lab actually dedicated at Pasteur Institute, dedicated for, dedicated for working at venom from scorpions, from snakes. So, and they are doing really a wonderful job. So, without any further ado, so I would like to welcome Mara Morgen, who will be presenting today a talk about snake venom as an anti-cancer therapeutic agent from 2D culture to 3D culture models. Thank you very much, and the floor is yours.
Thank you, Hideh.